Okay. Good morning. We continue with the course of pharmacology and physiology of neuropsychiatric disorders. And first, again, some small organizational issues. I'm sorry for this, but I will have to suggest another small change in the topics. This was the plan that we agreed before, and I just noticed that we omitted one very interesting topic, which I should be covered. It's interaction between endogenous opioids and monoamines, which I totally forget about last time. And this is the schedule how I recommend it now. This last class that we have, it was interaction between amino acids and monoamines. Today we will speak interaction between opioids and monoamines. And two last classes, it will be corticosteroids and growth factors. In addition, we have extra topics, which to a little bit less, to my opinion, a little bit less significant, but also we will try to talk about them. Extra topic one is adenosine monoamine interaction, and the second topic, which was based on very fresh results from my lab, which are very interested, is the nitric oxide monoamine interaction. And we will put these extra topics in the end of the lessons, because usually we always finish a little bit before the time. Today we will speak about monoamines and opioids. And some of you who were present yesterday on my, it was yesterday? No, it was 2 days ago, on my seminar, on my department seminar, it was partially about this. So for these people, it will be a little bit repetition. And at the end, if we will have time, and I think we will have time, we will start to speak about those adenosine. But before we start with this topic, I would like to come back a little bit to the previous topics, to about amino acid monoamine interaction, because one very important slide summarizing the interaction between serotonin and glutamate. You and me were tired, and we did it a little bit in the unfocused way. So I suggest briefly to come back to it. I will show you soon illustration from this paper from Frontiers in Integrative Neuroscience, Telade and colleagues. And I uploaded this paper to the 2 days to the 2 days directory. It's not mandatory to read it, and somebody who will wish to read this paper in depth, it will be not easy work, because it's very complicated, long, and review study. But one illustration from this, we will briefly go through it, because it summarizes what we talked about last time about interaction between raphe nucleus and prefrontal cortex. As we started in this course, for monoamine neurons, and especially for serotonergic neurons, very important issue is autoregulation. And we started with that serotonergic neurons autoregulate themselves via 5-HT1A autoreceptor, which expressed on cell bodies and dendrites. You see this is neuron. It's sent part of the branch of its axons innervates its the own neuron, and this is mediated via. But this loop of autoregulation is called short loop autoregulation. It's short loop because it's on the level on one neuron and one brain structure. It's all within the dorsal raphe nucleus. But there is also existing long loop of autoregulation, and this long loop involves prefrontal cortex and pyramidal neurons in prefrontal cortex. The dorsal raphe nucleus neurons, they send axons to prefrontal cortex, And in prefrontal cortex, they innervate, they made synapse with pyramidal neurons. And there are two types of synapses. The most important, most common synapses on the axonal part on the axon of pyramidal neurons. And this synapses work through 5-HT1A autoreceptor. Not autoreceptor, in this case 5-HT1A heteroreceptor. And you remember, what is the physiological effect of 5-HT1A receptor? Who can tell me? Inhibitory. It's inhibitory receptor. In this case, we inhibit 5-HT neurons, inhibits pyramidal neurons in the prefrontal cortex, and prefrontal cortical neurons, they tonically stimulate dorsal raphe serotonin neurons. In this case, we also have self-inhibitory effect, but long loop self-inhibitory effect, which involves another neuron in another brain area. But the picture is much more complex. And 5-HT1A neurons, they also make excitatory synapses on prefrontal cortex. And these excitatory synapses usually located on the apical part of the dendritic tree. And they also make synapses on the interneurons, on GABA interneurons. And these synapses usually involve 5-HT2A autoreceptor, 5-HT2A receptors and 5-HT3 receptors, serotonergics. Both of them excitatory receptors. The means the summary effect will be also inhibitory, because we make excitation on the inhibition, and finally we get inhibition of the cortical neurons, which leads to deactivation of dorsal raphe nucleus. Probably ketamine, antidepressant-like effect of ketamine works somewhere in this puzzle, but we still don't know how it works. Some of you may choose to make theoretical modeling work, how this puzzle of inhibitory and excitatory synapses may lead to increase in the net neurotransmission. Any questions about this monoamine glutamate interactions before I go to the main topic of today? No? Okay, let's continue to our main topic of today. It will be interaction between opioids and monoamines. In the left, what we see here is called the tree of knowledge. Etzadat. It's one where I think from biblical legends, it's maybe the most known one. Most of the people who don't know that much from the biblical text, but I believe everybody heard about the tree of knowledge. In Europe, most common to think, the most common idea that the tree of knowledge is the apple tree. And why it is, it's because of the very famous painting of Lucas, German painter from 16th century, Lucas the Older. And why he painted apple, more probably it was the most known fruit tree in the Europe at that time. Jewish sages, they never remind apple. They have different opinions of what was actually the tree of knowledge, but Russia, the most like popular commentator of biblical text, he states that Bible specifically did not want to tell us what was the tree of knowledge, because we will nobody will blame it. Otherwise, everybody will come to this poor tree and to come, you see all problems because of you. And as a neuropharmacologist, we have several candidates what can be tree of knowledge, what kind of plants brought to us put us between very difficult choices between good and evil. Two of them we already met, two very likely candidates to tree of knowledge we met in this course. One is coca, bespoke and product from cocaine, which gave to us, it's a It was actually first antidepressants. It's a precursor of all local analgesics. This is good, but evil. It's very bad and severe addiction to this drug. Another we already met in the previous class, I think, when we spoke about GABA, it's grapes. Those are good candidates to tree of knowledge. Also, alcohol, which produced from this, put us between choice of good and evil. And today we will speak about another very good candidate. It's Papaver somniferum, or Papaver. Is this beautiful poppy, very beautiful red flower. After this flower stopped to flourish, the fruit is forming. And fruit is such a how to take such a structure containing white semi-liquid matrix and semen. After this fruit gets dry, and its seeds, they're absolutely innocent. They used a lot in culinary. They used a lot in bakery. You see here the Papis strudel, where what I'm missing. What I'm missing here is very common dessert in our Central European region. So the seeds and the dry fruits are absolutely innocent. But if you take this white matrix from from this fruit and dry it out, you get what is called opium. And opium historically was consumed, usually most commonly by smoking, but you can also make tincture, you can also dissolve it in alcohol and consume it in this way. And this have very strong pain-killing and euphoric hedonic effect, this drug. Usually now it's very common. For a long time I was sure that that opium is originally from China, but it's not true. It's what is very interesting. Originally, it's European drug. It was probably discovered in ancient and classical Greece. This is a ancient Greek sculpture representing usage of smoking, usage of consumption of opium. This is real like opium milvan in English. Milvanin. Fossil. It's opium. It's a fossils from opium, real fossilated opium. Hippocrates wrote about analgesic and euphoric effect of opium. And with Alexander the Great, probably opium was brought to the east, to the south, to the South Asia, where it's become to be widely cultivated. Usually, about all drugs of abuse that we met before, the problem started when active compound was isolated. In case with opium, problem started before. When problem started, you see, this is the flag of Ost India Company. It was a British merchant company. It has a monopoly for trade in all British colonies in the Southeast Asia. But they not only had monopoly for trade, they actually owned huge lands with a lot of lands with millions of people living on these lands. And they started to cultivate cover in Bengal, what's in the eastern India, what is today known as Bangladesh. When they decided to sell it in China because it was nearby, it was huge population, so it was more likely and more easily marketed when you sell the opium. And it has drastic negative effect on Chinese society. If possible to say that it's destroyed the ancient Chinese empire. Very addicts becoming all parts of any parts of Chinese population become addicts, but more who people with a status and with the money. It's with governmental official and governmental officials, governmental bureaucracy. They were like core and pillar of the Chinese society, and at least more than half of them become heavily addicted to the opium, and China tried to prohibit and to stop this practice selling of the opium, but then England had two military intervention with forced Chinese to continue to sell opium, and it was very dramatic effect, and we stopped only in modern China. They stopped. It's a very sad side about the opium. And active compound of the opium, it's a morphine, he was isolated in 1803 by Friedrich Serturner, French chemist, and it was called after the Greek god of sleep and dreaming. Again, like with all psychoactive compounds, we get a good and evil. All painkillers, effective painkillers, they all come from morphine, and it's very necessary in the treatment. They make complicated surgeries possible which results in chronic pain after the they made possible to treat patients with wounds and burns. But unfortunately, immediately it created a big number of addicts because this substance not only pain killing, but it's also hedonic and addictive. And heroin was first artificial opioid, not coming from papaver, chemically synthesized. It was created to treat morphine addiction, and it treated morphine addiction very well because it was much more addictive. And it created much more dangerous, much more terrible addiction because it's not only more addictive, but it's also more toxic to the central nervous system. Methadone it's another artificial opioid, and it was created to treat heroin addiction. And more or less, it's also used as a legal substance, but not that much. It's more or less, the strategy was more or less successful. How the opioids act? They act through three type of receptors, so opioid receptors. And there are three types of opioid receptors, mu, kappa, and delta. They have pretty different functions. Activation of mu receptor is responsible to analgesic and euphoric effect of morphine. It's also believed that it's primarily responsible for addiction. Although delta also, unfortunately, also is responsible for addiction. Kappa, it's also receptor, has also analgesic effect, but very interesting. It has opposite effects. Have not euphoric. It has euphoric effect on the mood. And functionally, that receptor is very interesting one because it's involved in anxiety, stress response, and probably in depression. This is map of brain distribution of receptors. You see, for example, the delta receptor that we interested is pretty much responsible in the limbic area of the brain, but also in the ventral tegmental area and substantia nigra. This is a signal transduction mechanism for delta for all opioid. All opioid receptors, they have different effects, but they all GI protein coupled, very interesting. But like the serotonergic receptors, it can have very different electrophysiological effects. It may activate, in some cells, activate calcium channels. And as a result, it's have excitatory effect on postsynaptic neuron. But in some neurons, in some cells, it activate potassium channels and has inhibitory effect. Later, we will come and we will see really, we will see neurophysiological effect of delta opioid of opioid receptors. They indeed very interesting and sometimes can be opposite, can be from excitatory to inhibitory. It's a very good question. Oh, I had asked you before, very good question. Why, how can you explain that alkaloid from papaver has a so strong effect on the mammal central nervous system? We separated, in evolution, we separated papaver and we have common ancestor, but it was billion years ago. So how you explain that something from the plant has such strong effect on us? You heard about endogenous, you of course heard about endogenous opioids. Yes, and the answer is that actually these exogenous opioids, they have such a strong effect because they resemble some endogenous molecules. And these endogenous molecules, they got opioids, and they divided to three categories: endorphins, enkephalins, and endorphins. And enkephalins, they act predominantly, mostly, they have a little bit higher affinity to the mu opioid receptors, enkephalins to the delta, and endorphins to the kappa, but it's only relatively. All endogenous opioids, they act on all three types of more delta opioid receptor, on all three types of opioid receptors. Yes. Endocannabinoid. Yes, it's not connected. Yes, but it's not, and it's not opioid system. It's different system.
Opioid system creates much stronger
Maybe I cannot answer straight now, but I believe maybe because I believe that that opioid receptor much more widely expressed in the central nervous system. Widely expressed. We have I believe so, that we have much more. I never saw you see, because opioid receptors, you have them everywhere in the brain and pretty big amount. I'm not sure if cannabinoid receptor also that much expressed. Maybe it's one of the reasons. This is our endogenous opioid system, particularly the beta endorphin system. The neurons which are secreting beta endorphins, they are located in the arcuate nucleus. We will go today deeper to this brain area. And they projecting everywhere in the brain, and especially they project to the glutamatergic limbic areas of the brain and to the monoaminergic nuclei, to the locus coeruleus, to the raphe nucleus, and raphe nucleus and ventral segmental area, which suggests to us that it might be interaction between beta endorphin and glutamate, and between beta endorphin and monoamines. It was found in several studies that delta opioid activation of delta opioid receptors has antidepressant-like effect. Here you see Yudkevich study from 2005. They tried different doses of SNC80. SNC80 is a selective, highly selective agonist of delta opioid receptors, and they decrease immobility during the forced swim test, and they increase swimming and climbing. So it was pretty strong antidepressant-like effect. And this is our this is like this is our study. Did we speak already about open field test? This is an open field test. In this case, we treated rats chronically with SNC80, 3 milligram per kilogram per day. And we found that rats treated with SNC80, they have much more entrances to the central area of the open field. And what does it say if rats visited? Less anxious, exactly. It means that it has also anxiolytic effect. And now we will speak about neurophysiological effect of delta opioid receptor activation. And we will speak first about hippocampal glutamate neurons. And then we will speak about monoaminergic neurons. The first experiment with glutamatergic neurons we did in vitro conditions. We did it in primary cultures of hippocampal neurons. It's a method existing in our institute. We take newborn rats, isolate hippocampi, and then we feed the hippocampal neurons on primary culture, and they get saturated within 2 weeks, and you can within 1 more week you can work with them. You have glutamatergic neurons in primary culture. You can easily see it under microscope, and you can do intracellular electrophysiology. You can insert electrode straight to this neuron. And here we see the effect of SNC80 on hippocampal neurons. In low concentrations, it has inhibitory-like effect, but when you perfuse the neuron with a high concentration of SNC80, you have excitatory effect. Here we applied nautrindol, which is an antagonist of the same receptor, and it has opposite effect, opposite inhibitory effect. And if you pre-treat rat with nautrindol, and not rat, sorry, if you perfuse neuron first with nautrindol, after it you cannot anymore see the effect of SNC80. Then more or less based on our in vitro experiments, we get to conclusions that at least in hippocampal neurons, it has excitatory-like effect. And it's, yes. No, it's SNC80, the selective delta opioid receptor antagonist. Then we move to in vivo experiment. It's the same compound, SNC80, but in this case we administer it to the rat, and we recorded an individual hippocampal neuron in the anesthetized rat, and it was very surprising. The effect was opposite. We have inhibition, dose-dependent inhibition after SNC80. Then we tried chronic effect. It was acute effect. Here we administer SNC80 acutely. And here it was chronic effect, and was another surprise. Chronic effect was again excitatory, like we saw before in vitro conditions. We will later try to solve this puzzle a little bit later what it is, and we will first go to monoamine neurons. What we see is that we don't have primary culture of monoamine neurons. We don't know how it will work in vitro. We tried straight away in vivo conditions. Here we administer is a basal firing of serotonergic neurons. It's go down, firing is going down after administration of SNC80, and it doesn't recover after we administer antagonist, naltrexone. But if we give first antagonist, we have increase in firing. And if we give agonist after antagonist, this increase is going down. Dopamine, yes, is in vivo acute. I made a little bit mess. Sorry for this. And after chronic effect, we have again stimulation of neuronal firing activity. Yes, I did a little. It's also true for serotonin. I will bring it to the next lecture to show you. Sorry for this. With the serotonin and dopamine, we have same picture. We have inhibition after acute and stimulation after chronic. And this is probably what's happened. Why this? Why it's happening? It was pretty similar paradigm like we saw already with the trace amines, with trace amine associated receptor 1. We believe that effect of the receptor is excitatory, like we saw in vitro studies in individual cultivated neurons. But when you apply SNC acutely, we get increase in extracellular serotonin, and it was also shown in microdialysis studies, which I did not bring to this lecture, but I can show you later if you wish, or share with you papers. An increase in extracellular serotonin results in activation of serotonergic receptors. And serotonin usually extracellular serotonin leads to inhibition of 5-HT neurons and also of glutamate and dopamine neurons. And after chronic, after chronic administration of SNC, we probably have desensitization of serotonergic receptors. And after desensitization of serotonergic, we start to see this excitatory effect of the delta opioid receptor. Yes.
Can you say that there is no desensitization?
Desensitization. It's a very good question. Yes. It's from summary. We still don't know. We need to check it. First, we need to confirm it experimentally. After we confirm it, we will. It's not confirmed. It's still a hypothesis that. But from other side, we see the difference. We have inhibition after acute and we have stimulation after chronic. It must be some changes in the receptor sensitivity.
But there is obviously
it might be, or maybe with this compound, with delta opioids, and with trace amines, we do have the sensitization of 5-HT to A to C receptors, which we did not have with selective serotonin reuptake inhibitors.
Then sensitization is caused by the serotonin, right?
It's not only by serotonin. That might be also interaction between. Today, in the end of the today class, we will see totally another type how receptors can interact, and it can explain why you can get desensitization with one drug and you may not get it with another drug. Like my supervisor for master degree, Michael Newman of blessed memory, he used to tell when I saw some effect which doesn't contradict our papers, he told me, Elin, what to do? The animals did not read our papers. Now, let's, before we talk complete with about opioid system. Let's go to other side of the rope. קצה אחר של החוט. We were here. We saw in the interaction between opioids and dopamine neurons. We were here in hippocampus. We saw interaction between opioids and glutamate. We were here in the raphe nucleus, but we were not here in the place where opioids, at least some of the endogenous opioids, comes from the arcuate nucleus of the hypothalamus. And let's go quickly to this area, and we will see what's happened there. In this place of the brain, in the arcuate nucleus, there are neurons which are sensitive to leptin. And leptin is it's not brain neurotransmitter. It's a hormone which is synthesized by adipose, about our internal fat tissues, but by adipose tissues. And it's secreted by fat tissues, it's reached the brain, and it has effect on neurons in the arcuate nucleus. And there are two populations of neurons in this brain area. One has positive effect, and one has inhibitory response to leptin, and one has excitatory response to leptin. First, it was checked by Elias, and he used not electrophysiology, he used methods that's called C-force activation. C-force is proto-oncogen, and usually it's activated because of all kind of calcium signal mechanism. After chronic if you see excitable tissues like neurons, after chronic excitation, you have increase in C-force expression. What is good about this method is about C-force, you can check interaction with other molecular markers. And he found that neurons which express pro-amylocropsin, which is a precursor of beta endorphin, they have in POMC cells, C-force expression increased after leptin, and in other cells which are positive for neuropeptide Y, and neuropeptide Y it's not opioid, but it's another peptide, neuropeptide neurotransmitter, and leptin decrease C-force expression in NPY cells. We wanted to check it electrophysiologically. If there are indeed different types of neurons which respond differently to leptin. This is not easy to get to this brain area. We had to perform, after each experiment, to perform histological verification. If you don't miss it, it's 1/3 of millimeter, all this arcuate nucleus in the red. And we inserted our electrodes to this brain area, and we could find spontaneously active neurons, which have how it's called, this transaction potentials. They have bursts, I like bursts very much. And they have bursts, and they have pretty long and high amplitude action potentials. What happens when you apply leptin? Half of the neurons, leptin administration resulted in increase in the firing activity. And another half of the neurons that we observed, we almost did not found neurons with, it was almost no neurons with no response to leptin. Indeed, it was either stimulatory response or inhibitory response. And the stimulatory response is probably the POMC neuron and neuropeptide Y secreting neurons. And NPY response and NPY they have predominantly NPY neurons, they have negative response to the leptin. This is how it looks at in males and females. We saw it was not statistically different, but looks like that males, they have more massive, more prolonged effect to the leptin, both negative and positive. And this is basal activity of these neurons. Endorphin neurons, they have statistically similar between sexes, and NPY neurons, they have much more active in males than in females. You see that they respond positively to when you have high levels of leptin, when we're full of energy, when we don't need to search for the food. It means leptin, it's leptin and beta endorphins, they probably responsible for blockade of the appetite. And NPY other way around. In NPY we have other way around. Beta endorphin is responsible for food searching, and it's activated. I have always problem with this. Leptin, we have high levels of leptin when we have high level of energy, and we don't search for the food. It means activation, if leptin activate beta endorphin neurons, yes, they're responsible for the so-called consummatory phase of the reward. When we feel full, we don't search for the food, and we enjoy the current stage. NPY neurons, they probably responsible for the other, for the other phase, for preparatory stage, when we do need to search for the food. And probably this different activity, different type of activity between NPY and beta endorphin and neuropeptide 1 neurons between male and females, it might be somehow responsible for the different, why we have higher predisposition of the eating disorders like anorexia in females. Anorexia can be also in males, but it's much more rare disorder. And not only anorexia, we believe that this balance between beta endorphin and neuropeptide Y, it's, as I told, it's behind two types of the reward behavior. In every reward behavior, we have two phases. We enjoy mostly the consummatory phase when we reach the reward and consuming its reward, if it's food or drinks, it can be sexual activity, but there is also preparatory phase. And the preparatory phase also has its own reward. You know, some of you own cats. Some of you have had cats. Sorry, you have cats. You know, in cats, I think in cats this preparatory reward much more emphasized. Sometimes can be it's unfortunately from outside is impressed sometimes in cruelty. Cats, unlike all other, not only small cats, also big cats, unlike all other predators, they sometimes release the prey and attack again. Instead, just you catch the mice, instead to eat and to consume it, no, because there is also a very big reward in the fact of the reaching the reward. They can risk, as after all, mice can still escape, but they can even put it to the risk the consummation and to re-experience the preparation. And yes, sorry.
Or a bone and just knows how to catch it and give it back. But most of the time you see that they're getting prepared to attack it when I watch this and run over. And sometimes it just goes by it.
I believe in cats as very. And we believe, well, I'm a little bit now, I want to talk about this, but I feel a little bit disappointed. Yesterday, I get announced that my grant application, this topic was unfortunately rejected. I need to reapply for it. And why maybe today I'm a little speaking about it without enthusiasm, like I wanted to speak about this topic. And we believe that balance between beta endorphin and opioid, beta endorphin and non-opioid neuropeptide Y, probably in the behind the proper balance between preparation and consumption. And you must have balance between these between these two principles of the pleasure, because it might be that all our disorders, brain reward related disorders, then when you don't have proper balance. Let's take, for example, gambling addiction. Gamblers. They so focused on the preparatory phase. They so focused on this anticipation to the rewards that they don't need actual rewards. In gambler, even win be finally at one of the lucky days, gambler will win in casino big amount of money, he will immediately put it back on the stock. And other disorders, for example, drug addiction, it's a hyper consummatory, like opioid addiction. So what is interested to the patient, to the person, just to re-experience this consummation. Well, it's not always true, because also addicts, they enjoy the ritual of the preparation. Anyway, it's not very easy a puzzle. It's not, it's very simplified, but we believe that it might be a balance between beta endorphin mediated preparation and neuropeptide, between beta endorphin mediated consummation and neuropeptide Y mediated preparation, and it might be behind many disorders. And we also believe that it should be this balance between the beta endorphin and neuropeptide Y. They should be modulated by by glutamate and by monoamines. And I hope still to go to this direction. I think by this we finished with, as I told, we finished with to talk about any questions now about opioids, opioid monoamine interaction, but then I would like to go to another topic. Oh. Okay, then let's speak about this one of the extra topic one. It's adenosine monoamine interaction. I believe everybody heard about adenosine. There's no biologist, I think, first year undergrad biology student already heard about adenosine. Adenosine, it's binding stone of the DNA. It's adenosine is a nucleoside, and adenine, which is part of it, is a nucleotide. Also, when we see adenosine is it's our primary energy storing molecule. It's our biological battery. Because the adenosine they form, it can be phosphorylated, single phosphorylated, double phosphorylated, triple phosphorylated. In this phosphate to phosphate binds they can store high amount of energy, which can be reused after this after adenosine get defusilated. And adenosine is maybe also the adenosine is as a part of cyclic AMP is one of the most important signaling molecules, which we found it's already with the GIS couplet receptors. But adenosine is also a neurotransmitter, and you can ask me who can tell me where can we find adenosinergic neurons? Somebody heard about adenosinergic neurons? Actually, all neurons are adenosinergic. They all adenosinergic. We spoke about in my course, and I'm sure that you spoke about this in other courses. That adenosine is that storage of the neurotransmitter within the synaptic vesicles. It's an active process. You need to invest energy to store neurotransmitter within the synaptic vessels. Does not matter if it's monoamine neurotransmitter, if it's acetylcholine, if it's GABA or glutamate. And in order to store neurotransmitter, you need to use adenosine triphosphate. In all synaptic vessels, they have vesicular ATPases, which provide energy for the active transport of neurotransmitter to the synaptic vessels. And by some mechanism, which is still not completely known, adenosine is also get co-stored with all other neurotransmitters. It's possible that there is no neuron which releases some neurotransmitter and does not release some amount of adenosine together with this neurotransmitter. And it's not only like with trace amines, it's not only byproduct. It's also used, it has its own neurotransmittory meaning. And how we know it? Because there are adenosinergic adenosine receptors in the central nervous system. There are three types. One of the A1, A2, and A3. And A2, they have two subtypes, A2A and A2B. All of them, all adenosergic receptors, they G protein couplet. And A1 is GI. A1 and A3 is GI couplet. And A2 is GS couplet. It means all of them act on adenylyl class, either by inhibition or by excitation. We know that they are also found them in many areas of the brain. You found them in olfactory bulb, A2A and A1 receptors. You found them in the neocortex, in hippocampus, and in the thalamus. In the brain, we found mostly A1 and A2A receptors. A2B and A3, they mostly found in periphery, for example, in the heart. It's not end of the story. What is very interesting about these receptors, and it's not about this, but it's also one of the very, I promise you to speak about some new type of interaction between G protein couplet receptor, which we met before. It's a dimerization of the receptor. It was found. Some of these papers I uploaded to the course website. Who is interested in this, I really recommend to read it. It's, to my opinion, it's one of the most amazing things in G protein receptor pharmacology. A2A receptors, VTGS receptor, they can form a dimer, heterodimer with D1 dopamine receptor with a GI. And when the GS and GI receptors, they form dimer, we have totally different type of signal transmission. This heterodimer is already no more GI and no more GS. It's already coupled with JQ and has totally different way of signal transduction. And what is amazing, it's not only about adenosine and dopamine receptors. It's known that dopamine receptors can form heterodimers with other dopamine receptors. With many G protein, actually it's open to us to totally different pharmacology, which we never was thinking about it. Because we always used to believe that there is a receptor, ligand of this receptor and signal transduction mechanism. But now we know that different receptors, they can form homodimers and heterodimers. And after these changes, we have totally different way of signal transduction and totally different neurophysiological and neurochemical effects. And adenosine, very known, one of the E2 adenosinergic receptor agonist. It's antagonist. It's caffeine. I think it's much more. I think it's most most widely used psychostimulant. I think almost everybody use it. It's totally legal and widely used. And after I heard, I worked at the time in Netherlands in this contract research organization, and after I found this, by accident, I found this paper about dimerization, one of this paper about dimerization of the adenosine receptors, and I was totally amazed by it. I asked some time and resources, and to check it, if this dimerization of receptor has functional output in neurophysiological and neuropharmacological terms. And we performed this study. We checked the effect of administration of agonist and antagonist of D2 receptor and A2A receptor, adenosine and dopamine receptors. Here you see microdialysis study, because dimerization was found first with the dopamine receptors. We checked how the coactive blockade of these receptors results in extracellular dopamine levels. First, we administer Tourette's vehicle or antagonist of A2A receptor. It did not have effect by its own. And 1 hour thereafter, we administered to animal haloperidol. Haloperidol, it's typical antipsychotic drugs, and it's D2 receptor blocker. Because D2 dopamine receptor is an autoreceptor regulating dopamine, auto-regulating activity of dopaminergic neurons. We get increase in dopamine level, both in cortex and in nucleus accumbens. Yes. D2. D2 and A2. Sorry. Yes, you're right. I will correct it. And what we see when we pre-treat with ZM, with antagonist of A2A receptor, the effect of D2 receptor on dopamine release was potentiated. What is good with microdialysis? And microdialysis from the same dialyzer that you collect, you can check many neurotransmitter and setup of our HPLC chromatography apparatus was set like this, that is co-analyzed dopamine and noradrenaline. Additionally, even did not think to analyze noradrenaline, but we saw that that this compound also initiate the release of extracellular noradrenaline levels, what we did not expect to see in the beginning, because there is no data on dimerization of dopamine, of adenosine and noradrenaline receptors. And we also checked it electrophysiologically. Here we see, on the left, you see dopamine neurons from the ventral segmental area. We first administered agonist of A2A receptor. It has a decreasing effect, and antagonist has an increasing effect. And same effect was in the noradrenaline neurons. And this is how we believe it works. Probably in prefrontal cortex, in nucleus accumbens, probably like it was found in previous studies, there is a dimerization, molecular interaction between adenosine and dopamine receptors. And it negatively regulates ventral segmental area neurons. But probably there is also dimerization between, no one yet checked it. Many times I plan to do it, I never still did it. If maybe somebody of you want to come to my lab to undertake this project, somebody who is knowing good molecular biology, I really would like to find molecular interaction between adenosine receptor and noradrenaline receptor, and why this interaction can be very interesting. Maybe you can tell me. Let's go to noradrenaline is responsible for the feeling of the energy. If we feel energy, if we feel ourselves energetic, full of energy, or not, or we feel ourselves otherwise very tired and fatigued, and we feel fatigue, it's mostly because of our brain noradrenaline transmission. And adenosine is responsible not to the feeling of the energy. Adenosine is connected to the actual level of energy. Because when you in a higher level of energy, adenosine mostly double or triple phosphorylated. When you have dephosphorylated adenosine, it's when you out of energy, more or less out of energy. I'm speaking now in very general, like speculative way. Maybe this interact where you should be important connection between actual level of energy and feeling of the energy. Who can tell me in which type of disorders it can be crucial? Also, but also in the feeding disorders. Why why sometimes you not feel in the terms of the feeding of energy like you actually are. So it's a promising topics, but unfortunately, it's a little, for some reasons, pretty little interest. And well, I can explain for which reasons little interest to adenosine acting compounds, because adenosine receptors, they not everywhere in the brain, they not only in the brain, they also expressed, for example, in the heart. They have very strong effect on the cardiac activity, and the side effects can be very severe and very dangerous. And why today's pharmacology drug development is reluctant from going to this direction. But once we will have new way of drug delivery, when we will be able to deliver active compound to the target places, then this hopefully will be back interest to this topic. I suggest the remaining time to dedicate to your questions. Yes, yes, please.
So,
This one?
no,
More?
how do you see that interaction between, like, maybe try to put adenosine,
No, electrophysiology. It's very fair comment. In electrophysiology, it's not actual interaction, just effect of adenosine on receptor activation and inhibition on catecholamine neuronal firing activity. It's worth also to check. Of course, it will be very interesting to check both of them in combination, D2 together with. Actually, we did that. Well, yeah, we applied morphine, which is which is agonist of D2 receptors in the end. No, but you're right. We didn't really check the interaction.
I like the compound cell.
No, in microdialysis we checked interaction. We checked both of them. Yes.
And he also examined other cholinergic receptors, such as B2S and B2Y.
Excuse me?
Other cholinergic receptors, B2X, Y. This for B2S and they can also manipulate some cholinergic activity.
It's very likely to do. I don't know. I never learned this topic. You would like to take this topic for the work? There are more cholinergic receptors. No, not only adenosine. Yes. Yes, more questions from the Zoom. We have 2 Zoom participants. Would you like to ask something? In the note, we can finish here today. I hope you don't mind that we finish a little bit earlier.